In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you all, dear students? I hope you all are doing well and you are in best of your health. Dear students, once again I welcome you in Pakistan International School Thai's virtual learning system. As you know, this is Pakistan International School Thai's virtual learning classes, and today's class is for subject biology, which is your most favorite, I know, and your grade 8th. Now you are in 9th junior, so it is very highly appreciated, and uh, I think you all feel proud on having 9th junior class. So we are on chapter number 4, in that we are studying cells and tissues, and this is the continuation of our previous lecture in which we have started cell organelles and cell organelles. We have seen before the nucleus, mitochondria and ribosomes. Now we are gonna see some more cell organelles, which are plastids, endoplasmic reticulum and Golgi apparatus. These are some small organs of the cells which perform a very specific role in the cells. So let's get started. So today's topic will be started with the structure and function of plastids. Dear students, plastids are only present in plant cells. So plastids are only found in plant cells. And these plastids are like, if you see this structure, this is plant cell, and if you see this kind of the structure, that is plastid, which is present in the plant cell. There are lots of other organelles which are present in the plant cell, specifically these plastids. So here we have seen this picture before, that in cell organelles, this is plant cell. So specifically some organelles are present which are only present in plant cell. For example, cell wall as well as plastids. Plastids are having different forms or structures and shapes and the functions also. So plastids are what? Actually, they are the membrane-bound organelles. They are present in like bounded by membranes, and they only occur in the cells of plants and photosynthetic protists. As we know that they are present in eukaryotic cells, not prokaryotic cells. What is the reason for that? Because they are having membrane. So that's why. And photosynthetic protists are also eukaryotes, for example, algae. So they are having membrane-bound nucleus and other membrane-bound organelles also, and the algae which do photosynthesis. Because when we see the types of plastids, so one type is that which is only used for what we can say for photosynthesis. So three types of plastids we have found. Number one, chloroplast, leucoplast, and chromoplasts. Chloroplast, chloro means green, the plast, the bodies. Leuco means colorless, and chromo means colored. So these are the organelles or the structures which are having the pigments which are specifically for different colors. For example, chloroplast, you can see here, they are green in color because they are having green pigment that is called chlorophyll. So if we see here one by one, this is the structure of chloroplast, because this is very important indeed, because it is required and very important main structure which is required for photosynthesis for every photosynthetic or green plants. So like mitochondria, that is also double membrane bounded structure. The outer membrane that is smooth like mitochondria, and inner membrane, it is also having some different structures. Inner membrane is having some sacs, those are called thylakoids. What sacs are these? You can see this is outer membrane and this is inner membrane, and this inner membrane is not uh, zigzag form, but they are just transformed into the sac-like structures. These are the sac-like structures. These sac-like structures, they are called thylakoids. So this one is thylakoid, and the stack of thylakoid, like they look like the coins, just they present one above the other, one above the other. This one is thylakoid like this one, and this is stacked completely. This is called granum. This whole will be called granum. In granum, there are lots of thylakoids, 20 to 25. Okay, and here you can see that this is granum, and when we talk about all these granum, so they are called grana. Grana is plural and granum is singular. There are some glucose molecules which are coming here and they are used for generating ATP. Okay, so here now we can see they like these grana, they are composed of chlorophyll. That's why they appear green and they are necessary for photosynthesis. Some other accessory pigments are also present in the grana with the chlorophyll, like chlorophyll A, B, and some carotenoids and some other associated pigments are there which help in the photosynthesis. These grana, they are present in a liquid, or we can say they float in the fluid-like substance that is called stroma. So these pigments are present in the thylakoids of the grana, so they do photosynthesis while accepting the photosynthesis is what? By the help of sunlight and chlorophyll, plants chlorophyll absorb this sunlight, and with the help of carbon dioxide and water, they produce glucose and oxygen and water. So chloroplasts are having very important function, that is photosynthesis. They do photosynthesis. Chloroplast, I have told you before that chloroplast, mitochondria and nucleus, they are having their own DNA and ribosomes. Okay, so chloroplast and mitochondria also have their own DNA and ribosomes in these organelles to do their own photosynthesis and producing energy and proteins. Okay, so you can see the structure properly, like these are grana, this one the stacks, and this is called, this is a connection between one granum to the other granum. This is called stroma lamellae. Stroma lamellae. Here is the fluid that is called stroma, and there is the inner membrane. This is outer membrane. This one is called thylakoid, and this is inner membrane space, like inner membrane space between the inner and outer membrane. I hope you understood it clearly. Its function is photosynthesis. Let's talk about the other type of plastid, that is chromoplast. Chromo means colored. So these are the bodies which are having the colored pigments, and they are associated with bright colors. So where the bright colors are present in the plant? Definitely in the flowers and some of the fruits also. So these chromoplasts are pigment containing bodies which are present in the bright colored parts of the plant. So those are flowers and some fruits and some different colors of vegetables also. So they are present in the petals, we know, and these petals, because they are bright colored, so the insects and other organisms are attracted towards the flowers for pollination. What is its function? Like, as we know that they are colored bodies, so its very important function is to help pollination and dispersal of fruits, like the animals or like bats, like squirrels, they eat the fruit of that and they spread or disperse the seeds of the fruits anywhere and the plant can grow from that. Those are called dispersal of fruits also, and as well as help for the pollination. Now we'll talk about leucoplast. Leuco means colorless, so they are the colorless pigmented bodies. Like they don't have any type of pigments over there, and very important function of that, they are the storehouses of the food. Like the food is like where the food is stored. For example, in the vegetables, especially underground, and in the fruits. So the leucoplasts are present because these plasts stores the starch. Starch is the preserved or stored food in the plants, and they store proteins and they store lipids. So we know that all the fruits and vegetables are rich in carbohydrates, proteins, lipids are there, as well as lots of vitamins and fibers are also there. So starch is a big molecule that is called macromolecule that is made by carbohydrate carbohydrates. So the large molecule of carbohydrate, that is starch, and that is the stored food of plants. Okay. So up to here, chloroplast, we have discussed a lot about that. I hope you understood it well. Now we'll talk about endoplasmic reticulum. Endo means inside, plasmic means plasma or cytoplasm, which is present in them. Reticulum, the connections which are formed in the cell, and they are also present where in eukaryotic cells. Definitely, if you see, this is animals eukaryotic cell, and if you see, this is mitochondria, these are lysosomes, and this is nucleus. And if you see, these all connections which are around the nucleus, they are called endoplasmic reticulum. Actually, endoplasmic reticulum is having specific function. We'll discuss after that. So what is that? That is actually the network or interconnected channels. So you can see these are the channels and network of like channels, and the channels function is to transport the material from inside to outside of the cells. Sometimes they move from here and they just move towards the cell membrane, plasma membrane, and they move the substances out. Sometimes they transfer the material from the nucleus to the Golgi apparatus. Sometimes they just move the material from nucleus to the cytoplasm. So they are having the different functions. So there are two types of endoplasmic reticulum. One is a rough and other is smooth. Rough endoplasmic reticulum means it appears rough from the outside. If you see, this appears rough and this appears smooth. So you can see this is rough endoplasmic reticulum and this is smooth endoplasmic reticulum, that is represented by RER and SER. This is nucleus. Nuclear envelope is around that. From nuclear envelope, these extensions are there. They are appear like jigsaw puzzles or which like puzzle, isn't it? You have to move from here and find out your way out of this membrane of from this channel, and then you can come here. Like if you move from here, then this way, then this way, then you can move out. So material from the nucleus, it moves out into the cytoplasm. So they appear from the nuclear pore. They are having the connection up to the cytoplasm. And sometimes they are having the connections towards the Golgi apparatus. What are these? I will talk about in the next lecture, inshallah. Okay, so why they appear rough? Because they are having lots of these dotted like structures, these dotted or grains like structures. They are called what we have studied before, ribosomes. See, these ribosomes are present great amount, which are not freely moving. Freely moving are also present in the cell, but some are just stuck with that endoplasmic reticulum. So that's why they are called rough endoplasmic reticulum, because it appears rough in appearance. It's very important function, because what's the ribosomal function? Its function is to protein synthesis. So these rough endoplasmic reticulum, not only they are having the function of channelize the substance towards the cytoplasm, but it also having the function of protein synthesis. They produce photosynthesis and protein synthesis. They do for protein synthesis and like transport protein out of the endoplasmic reticulum towards the cytoplasm. Okay, the next one is smooth endoplasmic reticulums. Or it is smooth because they don't have ribosomes on it, so its appearance is very smooth from outside. So what is the function of that? Its very important function is lipid metabolism. It does not have ribosomes, so they don't do protein synthesis, but they do lipid metabolism. Like lipids break down on, because lipid is the very big molecule of fatty acids, so it breaks down in the this smooth endoplasmic reticulum, because it is having some enzymes which break this down. So it also transport the materials from one part of the cell to another part. And very important function also, it detoxifies the harmful chemical substances that enter into the cell, because it is having some sort of enzymes which can detoxify. Detoxify means if those substances are harmful, so they break it down with their enzymes, and then the substances will removed out of the cell. Okay, so Golgi apparatus. Okay, so next we will talk about Golgi apparatus. So this Golgi apparatus is having the connection with rough or smooth endoplasmic reticulum. If you see here, this is rough endoplasmic reticulum, and it is having the connection with the Golgi apparatus. This is endoplasmic reticulum. Some vesicle and some vesicles are produced from here, and they move towards the Golgi apparatus. Actually, what the Golgi apparatus are? Golgi apparatus, it was discovered by Camillo Golgi, and he was awarded for the Nobel Prize in the work of physiology, because he also discovered the function of Golgi apparatus. So physiology means the study of the functions of different uh, mm, organs or organelles inside the body. And medicines also, he also worked on the medicines in 1906. So if you see the set of flattened sacs, those sacs are called cisternae. If you see this is Golgi apparatus or complete, that is called Golgi complex or Golgi bodies. So this is the flattened sacs. If you have seen in chloroplast, there are also the rounded sacs, flattened, but they are smaller, but they are some larger flattened sacs. They appeared like on one another and stacks, they form the stack like structure, and these sacs are called cisternae. These are called cisternae, and in this set many cisternae are stacked one over the other, and complete this part is said to be Golgi apparatus. This is plasma membrane of the cell. So what will happen? This is endoplasmic reticulum. First, we'll talk about its location. It is located in plants and animal cells, but in eukaryotic cells. Here you can also see endoplasmic reticulum, and you will see these vesicles are going towards the Golgi apparatus. So what is its function? Actually, it modifies the molecules coming from rough endoplasmic reticulum and packs them into smaller membrane-bound sacs called Golgi apparatus. If you see here, these vesicles are coming from the rough endoplasmic reticulum and they are modified here and become the part of Golgi apparatus. You can see here. Then they move inside there in the cisternae and they produce, modifies means there are certain enzymes which just detoxify them, break them out, all the molecules, and they remove that vesicle outside. When they remove the vesicles outside, these vesicles can just burst out of this cell wall or cell membrane. So this is called exocytosis. Exo means outside, cytosis means cells. We'll talk about this procedure in some lectures. So exocytosis, exo means outside from exit, and cytosis means cells, like it breaks, comes out of the cells by breaking this cell membrane or this cell, and it removes the wasted material. Some of the vesicles just move inward inside the cells, and they are like they moved into the cytoplasm somewhere for its internal use. These sacs can be transported to various locations in the cell as well as to exterior in the form of excretion. So like it does the excretion like that. Okay, dear students, this is enough, I think so, for today to understand all and digest and absorb all such type of information. Okay, so today we are gonna discuss some of we can say the questions which is on your exercise, that is the page number from your exercise, that page number is 84, and that is on page number 84, this exercise. And now we are going to discuss the questions related to our the topics which we have studied yet. First of all, in there, the MCQs we will inshallah will do in the last of this chapter. Then it's understanding the concept and short questions. So let's talk about one by one. So here, number one is explain the function of cell membrane. Dear students, you all have to do this, all work on your notebooks so that you can make your own notes, and that will be easier for you to remember or learn that. So cell membrane, you have checked this we have seen in some previous lectures, cell membrane. So you have to just write its function. Its function of cell membrane, go to the topic of cell membrane and you will find out over there the functions of cell membrane. Then describe the structure of cell wall. So I mentioned separately the structure of cell wall. In that you will also write prokaryotic and fungi cell wall is made up of chitin and whatever the prokaryotes cell membrane is made up of. Cell wall is made up of peptidoglycan. So you have to write this one, as well as you have to write primary, secondary, volume, plasmodesmata. All those two paragraphs will come in this function structure of cell wall. Remember one thing, that what is asked in the question, you have to just write to the point answer. Okay, discuss the nucleus structure and the function. It means all the things of nucleus, about relating the structure, nuclear membrane, nuclear pore, nuclear plasma. These all will come, the chromosomes, all, and then nucleolus. So there is a function of every topic of this with its structure. So you have to write all of this about. Describe the structure and function of endoplasmic reticulum. We have discussed today. If we have to do write the structure. In structure, we will just talk about the two types of endoplasmic reticulum also, and the functions also. Then describe the formation and functions of lysosomes. Actually, we didn't discuss that right now. So inshallah, in a few, maybe inshallah in the next lecture, we will talk about formation and function of lysosomes. So we will discuss it inshallah. Then explain what would happen when a plant and animal cell is placed in hypertonic solution. Actually, um, as you know that because this term is very short, so we don't have this question or these topics of passage of the material. This is not included in your chapter number 4. So that's why this question we will discuss in 9th senior inshallah in the next session, not right now, but we'll skip here in this class, but not in 9th senior. In the next session inshallah we will just continue the topic as well as we'll do this question. Describe the internal structure of chloroplast and compare it with mitochondria. Very, very interesting and important. Internal structure, like it is having outer wall, outer membrane that is smooth for both mitochondrion and chloroplast, as well as its internal structure, its stroma, its cytosol. All you have to mention. Internal structure of chloroplast, first describe this and then compare it with that of mitochondria. Not important to just write in columns. You can write in paragraphs like comparison of chloroplast with mitochondria. So first discuss the internal structure of chloroplast, then compare it with mitochondria. Like mitochondria is having cristae, it is having cisternae. Mitochondria is having zigzag structure. On that, the proteins molecules are there for what aerobic respiration. It is having for photosynthesis, and like that you ought to all like function and structure when you compare that. Okay, then when you send me the homework, then we will inshallah just discuss it again. And if you have any query, any question regarding the lecture, you can just contact me in Zoom meeting right after your lecture. So short questions are also okay. There are some other understanding the concepts, but these are we didn't discuss the topics yet. So when we discuss the topic, then we'll continue the questions also. Short questions related to your topic, that is the state, the cell theory. You have to just write the Thayer and Sean gave the modern concept of cell theory, and you have to write the 3 concepts of that. Number 2, write the functions of leucoplast and chromoplast. What are the functions of them? So you have to write the functions of both of them. We have discussed in this lecture. Then the third question is not related to the topic because the topic are skipped in this term. We will discuss them in 9th senior, and then we'll do this question. And number 4 also. So here you have to do just only question number 1 and 2. The terms to know from chapter 84, we will inshallah discuss in the next lecture. Dear students, wish you all the best. I hope you found it very interesting and informative, as well as you understood it a lot. We'll inshallah meet in the next lecture or in the Zoom meeting. Till then, Allah Hafiz. Assalamu alaikum wa rahmatullahi wa barakatuh.